Thank you very much, and good morning. So what I'm going to do, I think that, I was asked to participate because what I do is I'm a bone marrow transplant physician. I take care of predominantly children who have a variety of life-threatening conditions. And also because of the work that I do in umbilical cord blood transplant, I think, what I've heard over and over is such a natural therapy. So it's kind of funny how, kind of things come around in some of the slides that you showed previously. But also, I deal with desperately ill patients, particularly children, and I hear about these, new therapies all the time, and trying to compare what I do versus what's available out there for some nominal fee. Next slide, please. Unless I can do it. So as all of you know, there's a variety of different diseases for which these people are quite desperate, and what they're faced with is a conventional therapy and sometimes palliative care. And unfortunately, that does not give many of them much hope. Next, please. So then, therefore, if there's not much hope with the conventional therapy, then they look for something experimental next, or they look for something that perhaps is a new treatment in the overseas clinic. And I only say overseas clinic because that's where many of these things occur. But as Alta has just shown you, you don't have to go too far to get some of these, quote, novel treatments that are currently unapproved. Next slide. So as an example of something that I do is to take care of some really awful diseases such as Epidermolysis Bullosa. And I'm only using this as an example of what desperate people are looking for and some of the sort of experiences I've had over the past couple of years dealing with such types of patients and families. And what we're going to do is talk about how, as a backdrop for the rest of the day's discussion, is, what does it look like in the field when you're dealing with a regulated therapeutic and going through all the regulatory processes. And I'll try to give you an idea for those of you that aren't familiar with what we have to do in order to get a trial forward. Next slide. So this awful, awful disease, it looks something like this. This is a 19-year-old who I met in Toronto and has this very severe recessive dystrophic epidermolysis bullosa. This adult patient would do anything at all, for any price. It doesn't matter. I could make up any kind of treatment and they would be there tomorrow. Next slide. And these are some of the quotes of the patients I take care of. The patient would say, I want to ride a bike, I want to play hockey, I want to swim, I want to wear flip-flops. They have very, very modest goals. And then a mother said she prayed every night that her child would die until she was given a chance for something. So again, you can only imagine what these families are going through, at least in certain circumstances, and this just sets the stage of what it's like. And it can be quite difficult being on the other side, trying to offer them a potential therapy when they have very little hope otherwise. Next slide. So one of the questions I was asked to address was, what is the evidence needed before offering a stem cell therapy to the public? Next slide. Well, there's a very long process by which at least most of us in this room probably would undergo such a treatment or develop such a treatment. And it begins first with an idea that's usually developed in the laboratory. And although in this particular group of experiments, this at least provided us with the preclinical proof of concept data that would allow us then to get subsequently FDA approval. But I'm not going to go into the details. It just really is to show you it begins first with the proof of concept. Next slide. And so interestingly, in this first cell therapy that we tried, actually many different stem cell types, and it was only the CD150 population of bone marrow that actually resulted in some clinical improvement. And what I'm showing you here is that basically the untreated animal with epidermolysis bullosa has blisters on his paws, and you can, although you can't see very well, in the untreated skin, looking under by electron microscopy, you find none of the anchoring fibrils that are missing in this particular disease, and then you see them present after the treatment with hematopoietic stem cell populations. Next slide. So this information is then, this is just going further. And interestingly, some of you in the audience that may not be familiar with these types of therapies, isn't it quite interesting that we were able to get FDA approval only with 3 animals surviving? Not very much, you might say. But the alternative, it also depends upon what the disease is, how severe the disease is, and what the alternatives are. But what these 3 animals did was provide the proof of concept because we were able to correct the disease and nothing else had ever done so at that point in time. Next slide. This is the clinical trial that's developed. The details aren't important, but the importance of this slide is to show you that what we did is we mimicked the clinical based on the animals. And therefore, that's how we move this whole therapy forward. Again, this is really just showing you the idea of what a regulated therapeutic might have to go through in order to become a trial itself. Next slide. The other thing that we had to do was have to have an oversight committee. So this oversight committee provided not only our external review to actually say which patients could move forward, because, you can imagine these desperate patients would do anything. So therefore, I had an external body that actually reviewed every single patient, and they would tell us who was eligible and who was not. Because as the therapist, I wanted to help them. And yet I wasn't also myself being the unbiased participant in this particular desperate situation. They also provided the DSMB review that occurred after the therapies were employed. Next slide. This is the before and after pictures. The top is before, the bottom is after. Quite extraordinary. But what was interesting is that the medical community didn't believe any of it. So therefore, I had to go even further. So we enrolled more patients. It actually was quite negative in the initial responses. And yet, this is a regulated therapy, and yet the people that are doing some of the unregulated therapies that we'll be discussing today, if you don't publish or if you don't present, then you also don't get criticism. Next slide. Well, then we also went further. We looked at the skin over time, and you can see, I think you can appreciate perhaps not because of the lighting, is that over time you would see that red linear streak between the dermal-epidermal junction, which is the missing collagen 7 that increases over time that wasn't present initially. Next slide. We then did electron microscopy, and we proved that we could actually create anchoring fibrils as a result of the stem cell therapy. Next slide. And then more before and after pictures. You can see on the left-hand side is before and then after. Next. Then before and after, going from left to right. Next. And then this is the continued evolution of the therapy. For the first time, we're creating fingers that were none existed before. But each one of these steps actually requires another regulatory review. So it gives you an idea of the complexity and the difficulty, the expense that might go along with that. So again, this is what it's like doing a very high-risk cell therapy. Next slide. Next slide. So then we also still had no believers out there. The dermatologist said that I was a witch doctor and that I was, what's it like murdering children? So the response to me was quite negative. And so we had to go further. How do you go even further to actually prove this to the medical community? And so we had to come up with more objective measures. And so we created a blister test, where we exposed the skin to different 12 millimeters of mercury, and then measure over time how long it takes for a blister to occur. Well, to make a long story short, that what you see here, perhaps on the right-hand side, is that you have the donor who has a normal blister-forming test of 60 minutes. Father and mother who are obligate carriers of this autosomal recessive disease have something where in between, although they don't have a blistering disease themselves. And you see the patient pre-transplant at about 10 minutes created a blister. And then over time, you can see where it improves. In fact, at 6 months after the transplant, the patient's blister test was better than the mother's. So again, and now it's becoming much more objective, and then over time, it's now becoming more believable, and the community is actually beginning to accept it more and more. Next slide. So this just gives you the overview of the translational pipeline, going from basic research to clinical research, and then going from phase 1, 2, and 3 clinical trials. And this is a laborious, long, expensive process in order to get an approval for a stem cell therapy or any other cell therapy or any other biologic. Next slide. But one of the second questions I was asked was, what are the best basic steps and standards for testing and approving stem cell therapies? Well, again, it's going from preclinical concept to phase 1 testing, which is really looking only for the maximum tolerable dose and toxicities. And then phase 2 is looking for efficacy, and phase 3 is the, quote, proof, and perhaps a randomized trial. So the numbers of patients get larger and larger and larger, and the expense goes up and up and up. Next slide. But it's more than just doing the clinical trials themselves. For example, to do any of this stuff, we have to have a procedure for manufacturing these cell populations. So one of the questions I was asked is, do any of these stem cell therapies have FDA approval? And as far as I could tell, next slide, the first stem cell therapy that was approved was Hemacord, which is basically an umbilical cord blood unit that was licensed by the New York Blood Center. And this is the only one in the United States that I could find, and perhaps there's others, but this is the only cell therapy that I could see, or stem cell therapy that I could see, which has been FDA approved and licensed. Next slide. And then we find Prochymal, which is a mesenchymal stem cell stromal cell approval that was in Canada, not in the US. And these are the only two that I could find in North America. Next slide. But when you look at stem cell therapies and you just go on the internet and look at where they've been approved, the majority of them have been approved outside the U.S. And in fact, there's a disproportionate number in South Korea. But when you also look further, many of these are not really stem cell therapies. They're really something different than that. And I just highlight a few: allogeneic amniotic membrane, allogeneic bone itself, and autologous cultured epidermal cells and chondrocytes. Next slide. Well, CBR, FDA, strategic priorities are really to enhance safety, clearly. And so safety is the major goal. And rather than going through all this, which I'm sure we'll discuss later on, the safety is more than just doing the clinical trial. Next slide. The safety is also in actually the manufacturing of these products. And this is at the University of Minnesota, but you can see the types of certifications the building itself needs, in addition to providing core functions in order to make sure that we give a safe product to the patient. Next slide. So there's the equipment monitoring that's required. Next slide. There's materials management, things that you might take for granted, and just making sure that the supplies that are present, are current, that they're not out of date. The materials have to be logged, tracked, controlled, quarantined. And so again, all these things require time, cost, people, and place. Next slide. There's also the document control. There could be deviations in the manufacturing scheme. There's going to be label control, things that you might not, you might think are behind the scenes are really a big deal, and particularly if you're a small university laboratory. Next slide. There's the facility control, air quality handling, alarms, inventory management. Obviously, you need to make sure that the air flow that's going through the facility is clean, as you might expect it would need to be. Next slide. And then certainly I highlight a few things in quality assurance. There's product release review for products produced under INDs because there's certain release criteria. For example, the stem cells have to have certain markers on their surface. They have to be free of endotoxin. They have to be free of bacteria and fungal elements. Things you would hopefully take for granted, but may not always be the case in some of these unregulated products that's being given to the public. There also needs to be equipment validation and calibration. There needs to be materials management, as I mentioned previously. Next slide. The one thing that was also asked was which stem cell therapies are undergoing clinical trials are under IND. Well, when just looking at our clinicaltrials.gov, we found 4,831 studies found for stem cells. Now, I'm not saying that all those are really stem cells, but when you did the search, stem cells in clinicaltrials.gov, that's the number you got. And many people will consider that to be, quote, FDA approved. I've had a number of patients say, well, it's under clinicaltrials.gov, so it must be approved. So there's a lot of misconceptions out there as what people find what they want to find, or perhaps this way it's presented. Next slide. This gives you an example of one such study, adipose-derived stem cell therapy for autism. Now, I don't know much detail about that particular therapy, but I'm a little suspicious. Next slide. And then you find Stem Cell of America treats diabetes successfully. But what's also interesting is sort of the words that are being used, and perhaps you can't read this very well, but it will talk about, that's been used successfully. But it also then talks about other types of diseases for which it's also being used for things that, Alta just presented. So it actually moves from actually the major topic of the heading, and it goes into other therapies. And it's almost by association, so therefore it worked here, it must work here. Next slide. And what this occurred to me is that really these are debate tactics. And we have to think about all the debate tactics and perhaps use that in our own favor. For example, questioning the motives of the opponent. There's a collusion between the FDA and Big Pharma. We hear this about the vitamin industry all the time. They're just trying to prevent you from taking vitamins because of the fact that there is some major nefarious relationship between FDA and pharma. So therefore, it's sort of a preemptive strike. These people are going to tell you something, and in fact, as a way of, when you do say that, then say, see, I told you so. Citing irrelevant facts or logic. For example, all the cures are with bone marrow, and then they'll start talking about some other disease. So they're linking two different areas. So cord blood we know can cure leukemia, and therefore it must be able to cure spinal cord injury. That relationship seems a bit obvious to us, but is not obvious to the regular person on the street, particularly if you're desperate. There's the unqualified expert opinion. For example, the very first slide that I showed you, it was some doctors saying, I believe in stem cells, and he and his wife somehow have some type of ability to communicate that results in people then saying, see, the doctor says that's what's good. Motivation and justifies dishonest means. That is, means cure at all costs. I, as your doctor, I'm here to cure you. And so therefore, however I get to that, and my hope and desire, that justifies it. And then finally, playing on the long-held fantasies and fears. And one thing I heard with cord blood, because that's where most of my research interest is that they would receive information because they're pregnant, they receive information from these different companies, cord blood collection companies, and it would start off, as a good parent, you would want to do everything for your child. The alternative is you don't care. And therefore, that's a good way of marketing, I guess, perhaps. And setting overvalued credentials, either fact eligible, that's a good one, or FDA friendly, or registered with clinicaltrials.gov. Next slide. So going back to what desperate people will do and how they interpret things, well, with conventional therapy or palliative care, the message is you're giving up and I'm hopeless. Okay, so if I'm the patient, I don't want that one. Next slide. If I'm going for an experimental therapy, such as coming to me under an IND, they get the message very quickly that we're very upfront with, our goal is to find the toxicity profile. I'm defining the maximum tolerable dose. The unsaid message is, only if you meet the strict eligibility requirements can you be on my trial. And in fact, with the Epidermolysis Bullosa example, the trials on clinicaltrials.gov said that we were going to be enrolling only 10 patients in the original trial. I had people actually offering me $1 million to be one of the chosen 10. They were so desperate. It never occurred to me that that's how people would interpret it. I'm thinking 10 because that's the science said I should do 10, and then we'll reevaluate and move on, obviously, if it worked. And then also the unsaid message is that I'm a guinea pig. So again, when we're thinking about the marketing strategies, these are words that would be held against us. Next slide. And then the new treatment that's overseas or even locally. The message is that this is personal. There's the testimonials. There are people like me saying that this worked and it was great. And for the first time, I now have control in my life. I get to choose the therapy I want. The doctor doesn't choose for me because they meet eligibility. Next slide. So in summary, the primary care provider is focused on comfort, but it's too realistic, you're going to die. Or you have some major illness that I can't fix. The principal investigator like me, the fear is I'm always overselling, so therefore I'm trying to push back and saying I'm looking for toxicity. Also, the patients tell me I'm cherry picking the patients and therefore they have no control, and you're focused on side effects rather than curing me. But the overseas clinician is saying, I'm offering you hope. And these other people, are not connecting like, we would. And then there's also the sense of victimization such that it's held against us. Next slide. So in conclusion, lethal diseases and markedly debilitating disorders require desperate measures. Some countries see a financial opportunity, and so therefore may be a conflict of interest. Marketing strategies include standard debate tactics. And while regulations are needed, I have to ask the question, is it possible that they may be too burdensome? Perhaps a question for another day. Thank you.
Thank you, Dr. Wagner. Thank you. The next speaker is Timothy Caulfield from University of Alberta.